Start. So thank you everyone for having joined this webinar. Good afternoon, good morning, good evening, depending on where you are. My name is Rita, and today we are going to speak about new technologies to culture spheroids. This webinar is part of a series of webinars that we started last year. If you want to have a look on those webinars, you can go to our website. We have other topics as cell migration or wound healing. This webinar is going to be recorded and is going to be available too in the same place. Also, after how this webinar is going to be organized, we'll have a first session of where we are going to present our technology to culture spheroids, and after we are going to have a session of question and answers. So feel free to raise your hand and we will unmute your microphone and you can ask questions, or you can use the chat box to ask us questions. If still you don't want to use any of these ways to connect with us, you can reach us by email after. We will have our contacts at the end of the presentation. So feel free to come back to us in any of these via any of these ways. So who are going to be the speakers today? We are three. So, well, as I already said, my name is Rita and I'm the CTO of the company. I'm here with my colleague Patricia, who is a R&D project manager at 4Dcell. And we also have with us Stein. Stein is an R&D engineer and he's doing a PhD with us here at 4Dcell. And he's the person responsible for the development of this technology. So at this moment, you are wondering, where are we located? What do we do at 4Dcell? So we are a French-based company. We are located very close to Paris, and we have in our offices all the facilities to produce everything that we provide at 4Dcell. So which things do we provide at 4Dcell? That's the next question here. So at 4Dcell, we have an extensive expertise in microfabrication, and we use this expertise to provide 2D and 3D cell culture substrates that mimic the in vivo conditions. So with this, you can do experiments that are more faithful to what cells sense when they are in the human body. All of our technologies are quite simple to use, and they are ready to use. So when you get them on your own lab, you just need to put your cells and things will happen. Also, we try to develop our technologies to be adaptable to high throughput screening and to high content screening platforms, so that not only academic users can have them, but also pharma and other biotech companies can use these technologies that we develop here. We have a portfolio of products and technologies available on our website that you can just go there and check what we have. But we also develop things based on specific demands, so we can do custom development. Feel free to reach us if you think that any of the things that we develop here or any of our expertise can help you move forward with your research. At 4Dcell, we have been developing over the last 5 years many different cell culture substrates that enable the control of the cell microenvironment from 1D to 3D. Today, we are going to focus on 3D, on a 3D culture system, a spheroid, something to form spheroids. But in the past, we have developed things like micropatterns, which are basically chemically modified surfaces where you can seed your cells and they will acquire a certain shape. For instance, if you want to differentiate cells, it can be very useful because it gives them a shape that resembles the shape that they have in vivo. We can have things that are a bit more complex, like structured surfaces. In this case, we can have, for example, hydrogels or micropillar surfaces, where we can tune the stiffness of the substrate. At the same time, imagine that you want to study cell migration. We have two devices that can be used for that. For example, our microchannels and our cell confiner. Well, the microchannels enable to study migration in one direction. The cell confiner enables to study migration in two directions. both of these devices can also be used to study the nuclear deformation. If you use our microchannels that, for example, have constrictions on them and that mimic the natural barriers in the human body that cells have to go through when they migrate. And the same in the confiner, you can induce a higher degree of confinement and you can see what happens at the nuclear level. Going a bit on this line of thought, we have developed more recently a substrate that enables the formation of spheroids. And before going into that, my colleague Patricia is going to give you a little bit more information on how you form spheroids, why are they useful, what can you do with them, and so on and so forth. And after that, I will get back to you to explain a bit more this technology. So Patricia, it's your turn now.
Hi, everyone. So thanks, Rita. As Rita mentioned, so she's presented 4Dcell, and I'm going to switch over now to the more scientific part of the presentation. So what exactly are spheroids? Basically, spheroids are spontaneously formed aggregates of cells that can better mimic tissues in the body. Basically, when you have a typical cell culture, cells will be attached to flat surfaces. And when you have cells in a spheroid, they'll be attached to other cells into extracellular matrix, which is a lot more similar to what happens in the body. On the right, you have a picture of a typical spheroid. So you can see that it's a self-organized structure. You have cells here that are on the surface of the spheroid, and these cells will have a lot more access to oxygen and nutrients that are in the medium. And at the center, you will have cells that have less access to nutrients and oxygen. So in this case, if you have a large enough spheroid, the inner core of the spheroid can become necrotic and the cells will die. So you really have a gradient of oxygen between the center and the periphery, of the nutrients, and even of the state that the cells are in, if they're proliferating, if they're quiescent, or if they're necrotic. So why are we interested in using spheroids? Well, as I mentioned, they are better mimics of in vivo conditions. You have predominantly cell-cell interactions rather than the interactions that you would have between cells and tissue culture surfaces. It's been shown that the gene expression and biochemistry of cells is more similar in spheroids than to in vivo conditions than cells that are cultured on 2D surfaces. And you can get a lot of interesting things. For example, if you have spheroids that are made up of stem cells or differentiated cells, you can mimic very particular organs. So here you have an example of a small intestine organoid from a mouse. And you can see on the image on the right, you have a schematic of what a typical organoid would look like. So on the left, if you look at the organoid, you have these spherical structures. And actually at the tips of these spherical structures, you have the stem cells that are shown in green on the schematic. And these are very similar to what you find in tissues of, here you can see the villi. So in this case, this is a cross-section of an actual tissue. And you can see here at the bottom, you have the actual stem cells that are segregated to the bottom of the crypt. So you're recapitulating what you see here on these spherical structures of the organoid. As I mentioned, you can get very complex architectures in spheroids. I've already talked about having different layers of proliferative or necrotic cells. You have oxygen gradients. You can also have pH gradients, and you can have even gradients of different cell types, as I was just showing with the organoid. Spheroids are also known to better imitate tumors, in particular, the tumor resistance to anti-cancer drugs. So in a 2D cell culture, all of the cells are in the same environment. And so if you expose them to chemotherapy, all the cells will die. But in a tumor spheroid, you have different things like the oxygen gradients, the pH gradients, the cell-cell interactions. And some of these can contribute to protecting cells in the interior from the drugs that you're adding. And so you will be able to recapitulate, in some instances, drug resistance to chemotherapy, tumor resistance to chemotherapy. So this is a much better model to use to test drugs. So how are spheroids made? Basically, what you want to do is you want to seed cells in a way that the cells will attach to each other and not to the surface of your container. So one thing you can do is to use low attachment surfaces. So you can see a schematic here where I'm showing, and pink is the low attachment surface. So you would just seed your cells on this surface and you obtain aggregates of cells. As you can see here, they're not very, what's the word? They're polydispersed, so you have a lot of different sizes. And with times, the spheroids tend to aggregate together, and so you get larger and larger spheroids. One way to mitigate this is to use microwells, where you seed cells in individual wells. And so you only have one spheroid, but they will all be the same size. You can also use a technique where you seed your cells in a drop. So I'm showing this here. You can basically, what you do is you add little drops of cells in your medium on a surface, and then you flip the surface over. And then the cells by gravity will go to the bottom of the drop and they will form a spheroid. You can also do a very similar thing using the oil and water emulsion idea, where you add your spheroids, your cells in an aqueous solution, and you make droplets in an oil media, and you obtain these cells in little aqueous droplets, and they will form spheroids eventually. So both of these techniques are a little bit more finicky, obviously. And then you can also use forced floating, where you prevent the cells from attaching to the surface by essentially keeping them in suspension all the time. So basically here you would have something that's constantly stirring the solution. And again, you end up with quite a bit of polydispersity. Another technique would be magnetic levitation, where you load nanoparticles in cells and then you levitate them so that they aren't able to go to the bottom of your container. So all of these techniques have advantages and disadvantages that I mentioned, you know, the polydispersity and the difficulty to actually do the technique. And so now I'm going to pass the mic back to Rita, and she's going to tell you a little bit about the technique that we've developed at 4Dcell, so that hopefully we can avoid all of these problems.
So thank you, Patricia, for passing the word back to me. As she just mentioned, I'm going to now introduce our smart spheroid plates. Basically, what we have is a very simple system where we use an hydrogel substrate. We mold microwells into this substrate, and these microwells have the shape that you can see here in this scheme. So they have inclined walls, which means that when you seed cells here, they are directed towards the bottom of this well. At the same time, because the gel is repellent, so the cells cannot adhere to it, they are forced and form this aggregate. So you can see in this image here that this is the normal look of the spheroids that you can form with these type of structures. And let's now go one by one through the characteristics of this assay. Basically, there is a self-assembly of the spheroids with a regular size and shape, because we have all of these microwells organized side by side in a well of a multi-well plate. So you just seed your cells, and since the cells are homogeneous in your medium, they are going to go down in an homogeneous way. And since the microwells always have the same size, they end up having a regular size and shape. There is actually no loss of spheroids during medium exchange, because we have added a very particular feature to our microwells. In fact, at the bottom of the microwells, there is a contact point that I didn't mention in the beginning, but this contact point enables the spheroid to anchor down, which means that when you add medium, add drugs, exchange, do some changes of medium, the spheroid is not going to be aspirated. And this is a big added value, because this is one of the biggest drawbacks of other methods. Other methods is that you lose a lot of spheroids when you do such type of manipulations. Also, all the spheroids are at a predictable location. This means that you can follow one specific spheroid, and you can be sure that the spheroids don't switch between wells because they are anchored to the bottom of the microwell. On top of this, they are very easy to image and compatible with standard imaging systems. So all the substrate is an hydrogel, which is transparent. And since the spheroids are attached to the bottom of the microwell, to the contact point in this case, it means that they're all at the same level. So when you take your plate to the microscope and you want to image a certain spheroid or all your spheroids, you don't need to look for them because they are in suspension in the medium or something like that. The nature of the substrate, since it's a gel, an hydrogel, it is porous, and it means that nutrients can migrate not only from the top, but also from the sides towards the cell aggregate. In addition to this, since it's an hydrogel, we can tune the stiffness of the substrate, and by tuning the stiffness of the substrate, you can have an environment that is again closer to the in vivo environment. To finalize, and this is something that comes, it's very obvious when you look at the images that I'm just showing here. You can have quite high throughput with this type of structures, of assembly, let's say, because we can have many individual microwells per well in a multi-well plate. I will now detail this a bit more in the next slides. So this here, you can see the top view of a well of a multi-well plate, let's say. Here we have 91 microwells. We can do this in 6, 24, 12 well plates, and even 96. Here you see the typical dimensions of the structures that we have available. We will have more shapes available that can allow you to have the spheroids more spaced from each other. And here you see a cross-section of a well of a 24 well plate, where you see that it's what you get. It's simply the well with the hydrogel at the bottom, and then you just manipulate this well plate as you would manipulate any other well plate. Here you see an example of an actual product. It's a 6-well plate, and here is a zoom of the hydrogel, the microstructured hydrogel. And as I just mentioned, you can get this in 6, 12, or 24 well formats, either with a glass bottom and then at the center, with a glass bottom at the center and a plastic bottom on the sides, or either with a complete glass bottom. We have different plate formats available. Simultaneously to this, we are developing the molding on 96 well plates, so that you can have this in a more high throughput formats. Going now to one of the most special features of our plates. In this image, you can see the impact of having a contact point in the microwell before vigorous medium exchange and after vigorous medium exchange. So you see on your left that you have microwells with contact points, and on your right, you have microwells without contact points. And what you see in both sides is that when you have the contact points, you lose just a few spheroids if you are really harsh on the medium exchange. Okay, it's not like normal pipetting. It's really being a bit tough with your culture plate. And on your right, if you have microwells without a contact point, you end up losing quite a lot of spheroids when you do a vigorous medium exchange. And this is a proof that you end up having many more spheroids to analyze at the end. So you lose less cells in this case. So now I'm going to pass the word to Patricia again, who is going to detail a bit more the features that I've just mentioned before.
Okay, thanks, Rita. So yeah, I'm going to tell you a little bit about the type of things you can do with the spheroid plates. So here I'm showing the idea that basically the size of the spheroids really depends on only how many cells you add basically to your well. So here we have added an average of 1,000 cells per microwell, and we have a much smaller spheroid than when we added 30,000 cells per microwell. And so the relationship between the two is basically a cubic relationship, because you're looking at the number of cells will depend on the diameter of the spheroid will depend on the number of cells. And so if you add 8 times as many cells, you'll get a spheroid that's twice as large. And this is what you can see in this graph here. The more cells you add, the larger the diameter. We can also look at the lifetime of the spheroid. So the way spheroids form is there's a first phase with aggregation and compaction. So we have a little movie we'll show you that will demonstrate this. And then following this aggregation and compaction phase, you will have a proliferation and spheroid growth phase. So you can see this very nicely in the curve. So at 2 days, you already have a nicely compacted spheroid. At 3 days, it starts to grow a little bit. And then at 8 days and 10 days, you can see it growing even more. Here we have the little video showing the first phases of aggregation. It's going to loop back again. So you can see very nicely the spheroid forming. And then after 24 hours, you already have a very spherical structure. And this will keep growing with time. And you can see this very nicely in the movie. Another thing you can do is just test different cell types. So here at 4Dcell, we tested these 4 cell types. On the left, you have MSCs, so human mesenchymal stem cells, human primary fibroblasts, human breast cancer cells. In this case, they're MDA MB231, so a very common model for triple negative breast cancer. And on the right, we have mouse myoblasts, so muscle cells. And what's really interesting is that in each case, we seeded 3,000 cells per spheroid. We waited 72 hours, and you can see you have very different results based on just the phenotype of the cells. So how much they'll compact together and how much they'll already be proliferating after 72 hours. So we can already see very big differences between the different cell types. We can also do quite long-term cell culture. Here we're showing an experiment where we kept the spheroids for 35 days. So you can see that they keep growing with time. And they're quite stable. All you have to do is change the medium. And you can see in this case, we didn't lose any of the spheroids. And as Rita mentioned earlier, due to the nature of the hydrogel, you really get nutrient exchange all around the spheroids. So the spheroids can stay quite healthy. One question that you might be wondering, because we've been talking about the contact point so much, is whether you can actually harvest the spheroids afterwards. For example, you might want to harvest them and lyse them to do some Western blotting or something. And so you can. We've tested different ways to do this. One way is enzymatically by adding trypsin, for example. So if you add trypsin and then you rinse the gel, you can recuperate the spheroids. Or you can do mechanically. So in this case, you have to be quite vigorous with the pipette. So you have to go up and down several times over the hydrogel. And what happens is you end up ripping some of the spheroids out of the hydrogel. And you can see that you end up with these very nice spherical structures that are basically your spheroids, and you can pick them up. If you wanted even to just grab like a specific spheroid that's interesting for whatever application you have, you could approach it with a very fine pipette if you're dexterous enough, and you could even just pick up an individual spheroid of interest. So now I'm going to pass over the mic again to Rita, and she's going to tell you about a new product that we're going to be launching soon that we're very, very excited about.
Thank you, Patricia. Yes, we are definitely very excited about this new product, what we call the Smart Heart technology. And this is basically the same technology we are using to form the spheroids, but rather than being just a well, the well has a central pillar. And this enables the generation of 3D cardiac organoids by themselves. So I think the video speaks by itself. What you see here, it's an assembly of iPS-derived cardiomyocytes that are contracting spontaneously. Basically, we use the structure of this gel to measure many readouts as contractility. If we wouldn't have the central pillar, we wouldn't be able to measure the contractility force that we are able to measure. In this case, we are able to measure the beating rate, beating amplitude. We are able to image ionic exchanges. We are able to see cell organization, cell morphology, and many other readouts. Well, this is something that we are just releasing. We are quite excited about it, so feel free to come back to us to discuss it. As you see here, the organoid is beating. What we can say on the top of what I just mentioned is that the organoids, the cardiac organoids, start beating 1 day after, more or less, that the cells have been cultured. We can keep them for a long time in culture. And so far, all the tests that we have been doing are very, very positive, and we are really excited about this cardiac assay that, due to the nature of its shape, it's a bit closer to the shape of the human heart too, so it's much more meaningful. The readouts you can get from here are much more meaningful. So feel free to get back to us with questions about the spheroids assay or the Smart Heart technology. Here are our contacts, but just after this, we are going to open the question and answer